All right, welcome everyone. In this video, I want to take a look at another example where iteration using for loops is going to be really useful. So I want to take some look at contexts in data science where these things might be used. So we look at bootstrap distributions, which are really useful in statistical inference. I want to take a look at what is called the KNN problem, or the K nearest neighbors, and how we can use a for loop to find these things. And we'll see in what context might a KNN process be useful. So this is definitely an application from machine learning. So if you've taken a machine learning class, you might be familiar with this. If you're not familiar with this, I'll give you a background. And if you want to learn more about it, I would definitely recommend a whole course in machine learning. But we'll use a very standard data set, which is kind of really foundational, particularly for like cluster type of problems in machine learning, which is a data set of observations of different irises. So what we're looking at here are three iris species, the setosa, the versicolor, and the virginica. And we have some measurements on each of these. So the data set, each observation is going to be a different flower. And for that flower, we're going to measure the length of the petal, the width of the petal, the length of the sepal, and the width of the sepal as well. So you can see these are labeled a little bit in this plot over here. Okay, so the data set that we're going to be working with is called Iris. You can see some of the documentation about it, and you can run like $Iris if you want to learn more about this data set, but it contains 50 flowers from each of the three species. So that means it has 150 observations in total. This data set is in base R, so you don't need to install any package to use it, but you do need to load the data set first. So we're going to load the iris data set. We'll take a look at what's inside of it. And you can see these are the first 6 rows of the data frame. We've got 4 numeric variables, dimensions of the sepal and the petal. And then we've got a last column, which is telling us the species. And if we plot this data set, so here I'm just going to create a plot using ggplot where we've got petal width. So I'm just going to focus on 2 of the variables. Let's just focus on the petal. So we're going to focus on the petal length and the petal width. And what a data scientist might do is say, maybe I don't know what species, right? You can imagine using like an app on your phone. They have these apps where you can take pictures of flowers and then it tells you what type of flower or plant it is based on like a picture of the leaf or something like that. So you can imagine like you take a picture, that picture records some dimensions about the leaf, and then based on those dimensions, it tells you what species it thinks it is. So what we're going to do is like, let's imagine we just know the petal length and the petal width, just to simplify things. So if I know those 2 measurements, how could I predict what type of species this plant is? So we can take a look at a plot of this. So I've plotted the petal width on the horizontal axis, the petal length on the vertical axis. And as you might suspect, there's a positive association and it is extremely linear, which might be surprising. But you can see is as the width of the petal goes up, so does its length. That makes sense. But when we shade these points by species, we can see there's like 3 clusters of points. There's a cluster down here for the setosa. There's a cluster in the middle, the green one for the versicolor, and then there's the yellow ones that are the virginica. And let's assume we didn't know the coloring, right? So all we knew was that there was a whole bunch of points. It seems like pretty clear that there's a cluster of points over here that probably corresponds to one species, and then it wouldn't be necessarily clear how to split this up. This looks like one cluster, but it's actually containing two different species. So one way that you can kind of do this clustering is by using the KNN problem. So the K is nearest neighbor, and based on which other points are close to it, you identify all of those points together as coming from the same species. So you can imagine that looking at the petal width and the petal length, points that have relatively close, similar dimensions are probably similar species. So that's what this KNN algorithm does, is it's going to identify other observations in our data set that are close to it. And we assume that if these observations are close, they probably have something in common with each other. So that's motivating the KNN problem that we're going to take a look at. And so in the next couple of videos, we are going to analyze this, but we're going to restrict our analysis to just working with the petal length and the petal widths. So we're going to take this iris data frame and look at only columns 3 and column 4 from that. We're going to keep all of the rows, but this code over here is doing a couple of things. It's pulling off just the third and fourth column, so just the petal width and the petal length. It's going to drop all of the other columns. And we're also going to rescale our values. And the reason why I'm rescaling these values is because the petal length overall is a bigger value than the petal widths are. And sometimes when you have different features in your model that are on different scales, it might put more emphasis, let's say, on the petal length than it does on the petal width. And if I want to weigh these two features evenly, then I want to rescale their values so they're on similar scales. So that's what this scaling function does. And if you want to take a look at what comes out of it, we just have two variables, petal length and petal width. These values are negative in this case. We shouldn't have negative lengths and widths. So these are not actually lengths and widths, but these are like pretty much like z-scores of the values. So this is telling me like how far away from the mean was the length of this flower, how far away, in this case, below the mean in terms of how many standard deviations away from the mean are each of these values. So now you can imagine we're given petal lengths and petal widths by their corresponding z-scores, more or less. That's what's happening here. So in the next code cells, we're going to use this rescaled data set to find the nearest neighbors for each point. But we'll pick that up in the next video.